Biotechnology 100 quiz we will discuss. So the first question is Unit 1 Biochemistry. Enzyme catalyzed hydrolysis of protein produces amino acids of the form. Which form it produces? It generally produces L form. We have two kinds of forms. One is dextrorotatory and levorotatory. But in our body, we will have L form, levorotatory. Okay, but in case of glucose, we have dextrorotatory, D glucose, not L glucose. But in amino acid case, it is L amino acids. Okay, so the answer is D. Enzyme catalyzed hydrolysis of protein produces amino acids of the form which type? L types, because in our system we don't have enzyme which can catalyze the amino acids which are which form? D form. Okay, so next questions. All the following compounds are intermediates of TCA cycle or tricarboxylic acid cycle, and this TCA cycle is happened inside the cell of an organism, inside the cell and inside the mitochondria of a organism, which is double membrane structures, and inside this TCA cycle occurs, tricarboxylic acid cycles. In this way it will work. Okay, so it is happen, but the pyruvate is not the part of the TCA cycle. It is the part of glycolysis which happen inside the cell, but not in mitochondria, but in cell cytoplasm. If this is the mitochondria, this is the cytoplasm, then the glycolysis happens in cytoplasm. Okay, so pyruvate is not the part of TCA or tricarboxylic acid cycle. Then identify the correct molecules which controls the biosynthesis of proteins in living organisms. So this is a relations with operant models or the switch on or switch off of the protein synthesis, all these things. So the biosynthesis of proteins is really controlled by mRNA or RNA. All means RNA is having three types. One is tRNA, one is mRNA, one is ribosomal RNAs. All these RNAs are involved in protein synthesis, and they actually decides what should be the protein, which sequence should be followed in proteins. All these things are present and controlled by RNAs. Then, so right answer is C. Then transaminase activity needs the coenzyme. Which enzyme? It requires B6PO4. This enzyme is required. Then next questions will go here. The fifth one, a mitochondrial marker enzyme is what? Succinic dehydrogenase. What is the answer? Succinic dehydrogenase. Then sixth one, which mineral element controls the activity of nitrate reductase? This is also one enzyme, nitrate reductase, and it needs one element called MO, or what is the molybdenum, MO. Then enzyme inhibition caused by a substance resembling substrate molecule is competitive inhibition. So if a inhibitor which inhibits the enzyme catalysis process, and that inhibitor resembles what substrate molecules, then that kind of inhibition is called what competitive inhibitions, because the inhibitor is competing against the substrate, so that the substrate is not capable to bind with enzymes. So this kind of inhibition is called competitive inhibition. Then the Michaelis constant Km is what? It is in numerical equal to the substrate concentrations that gives half maximal velocity. So suppose we are calculating the rate of a reactions, and if the substrate concentrations at which the rate of the reactions reach to the half of the total velocity, suppose it is the V max and it is the half of the V max, half of V max. So in this time the total number of substrate concentration, the total substrate concentration is equal to the kinetic constants. You see, Michaelis maintain constants. So whatever the substrate concentration at which the rate of reaction reaches to half of the velocity, maximum velocity, that substrate concentration is called what? Michaelis maintains constant. Okay, so the right answer is D. So the Km or Michaelis constant is Km, which is equal to numerically equal to the substrate concentration that gives half maximal velocity. The next question is oxidation of which substance in the body yields the most calories? So if we understand, we have lipids. Lipids gives more energy as compared to other compound present in our body. Okay, so lipid is the right answer. It gives how much calories? Most, it is almost double to the energy given by whom? Glucose or proteins. Okay, so right answer is lipids. Then alpha D glucose and beta D glucose are what? Anomers. Okay, these are quite similar but have little difference in their spatial arrangement. Chemically both are same, but in their spatial arrangement of the molecules they have some differences. That's why they are called anomers. Then phenylalanine is the precursor of what? Tyrosine. It is a precursor of tyrosines. Then an uncoupler of oxidative phosphorylation such as dinitrophenol is what? It inhibits the electron transport and ATP synthesis. So these uncouplers of oxidative phosphorylations is a poison which is called dinitrophenol, and if someone will take this dinitrophenol will die immediately because it inhibits the synthesis of ATP at mitochondria. So by uncoupling the electron transport chain, whatever the electron transport chain is present in the mitochondrial membrane, it will uncouple that compounds, by which what will happen? The ATP synthesis will be paused or the poison will kill the organisms. Then next question is 13th, which of the following is a substrate for aldolase activity in glycolytic pathway? So in glycolytic pathway, fructose 1,6-bisphosphate is a substrate for aldolase activity. Okay. Then in protein structures, the alpha helix and beta sheets are example of secondary structures. These are secondary structures. See this sequence of amino acids. Suppose this is a sequence of amino acids, and this is called primary structures. This is called primary structures. But when these primary structures will fold like this, by having some bond in between, then this kind of structure is called secondary structures. And we have two different types of secondary structures. One is alpha helix and another one is beta helix. So these are nothing but secondary structures. Next questions. The number of helices present in a collagen molecules is not more than 3. So right answer is 3. Then unsaturated fatty acid is known as essential fatty acids because we or our body cannot synthesize this not amino acids, fatty acids. This essential fatty acids we cannot synthesize in our body, and it is so essential fatty acids we cannot produce, we need it from outside. These essential fatty acids are unsaturated fatty acid. It means it has double bond inside the chain. In between the carbon carbon molecule atoms there are double bonds. That's why it is called unsaturated fatty acids, because it does not have sufficient number of hydrogen within it. That's why it is called unsaturated fatty acids. Okay. Next, for finding out aromatic amino acids, we do what? Xanthoproetic test in proteins. If a protein is having aromatic groups in the amino acids, we can do xanthoproetic test to know. Okay. Next questions. The half-life of antibody protein is how much? Not more than 3 weeks. Half-life of any antibody. Then beta oxidations of odd carbon fatty acid chain produces what? Propionyl coenzyme A. Beta oxidations of odd carbon fatty acid chain, or it is also called digestions of fats. Beta oxidation is also called as what? Digestions of fats. So in if a fatty acid is having odd carbon numbers, then there is release of propionyl coenzyme A. Next, the general formula of monosaccharide is what? CnH2nOn. If carbon number is 1, then hydrogen number will be 2 and oxygen number will be 1. This is the ratio. So right answer is A. Then next question 21. So it is from molecular biology and genetic engineering. So translation results in the formation of what? In translations we get a protein molecules. Okay, when DNA produces mRNA, this is called transcription, and when mRNA produces protein, this is called translation. Okay, next questions. In the lac operon concept, which of the following is a protein? See, in lac operon there are different parts. There is on operator, inducers, all these things, and there is also a repressor part which acts as a switch. When we switch on TV or light, like that, there is a switch kind of things which is also called as repressor, and it is nothing but a protein in the cell. This has happened in E. coli or bacterial cells. Then Another name for reverse transcriptase is RNA dependent DNA polymerase. RNA dependent DNA polymerase, and this enzyme or reverse transcriptase is responsible to produce what? DNA from mRNA or RNA. Okay, it will produce DNA from RNA. Okay, next question is 24. Nucleic acid show strong absorption at one of the wavelength. So we know different molecules have different observation capacity: protein, glucose, nucleic acids, and in a certain length, certain wavelength, they have highest or strong absorptions, and that wavelength is nothing but how much? 260 nanometer. Okay, 260 nanometer. Then next question: template directed DNA synthesis occurs in the following except growth of RNA tumor viruses. So template directed DNA synthesis occurs in all the following in the except where it is not happening. It is not happening in growth of RNA tumor viruses. Then a gene is what? An instruction for making a protein molecule. It is not but it nothing but an instructions in a different codes, A, T, G, C, G, C, E, C, like that. So it is an instruction for making a protein molecules. Then all the followings are naturally occurring nucleotides except cyclic AMP. These are all nucleotides. What? ATP, DNA, inosines, monophosphates. All the followings are naturally occurring nucleotides, but cyclic AMP is not the naturally occurring nucleotides. It is responsible for what? Transfer of messages or communications between different cells. Cyclic AMP. Then next questions is 28. The genetic code operates through what? Genetic codes operates through nucleotide sequence of mRNA. Okay, it cannot directly produce the protein, but it will produce the protein through mRNA. So the answer is C. Then 29. Proto-oncogenes are present in where? Healthy human cells. See, these genes are not the genes responsible for what? Means producing the cancer. If they are present, then cancer will not happen. But if these proto-oncogenes are converted into oncogenes, then that will be causing what? Cancer. That will be reason for cancer. But naturally these proto-oncogenes are not cancerous in nature if they are present in proto-oncogene state. But when they are converted to oncogenes, then they will call cancerous. And then the right answer is healthy human cells. You will find proto-oncogenes. Okay. Then 30th, each artificial chromosomes, or it is also called as what? YAC, is used to amplify DNA sequences of the size what? 1000 kilobytes. Okay, we can do what? We can amplify DNA sequences of the which size? Amplifications, or we can generate more and more numbers of DNA sequence having size of what? 1000 kilobase pairs. Okay. Then DNA fingerprinting is based on the presence of DNA of what? VNTRs, or called variable number of tandem repeats, and this part is present in a single person in, and there is no similarity between these VNTRs with other organisms. It means we can say a profile, we can generate a profile of different persons and recognize a particular persons related to that VNTRs. So these VNTRs are not equal to any other persons. So this DNA fingerprinting is done to identify criminals or any other activities we can do, but these VNTDRs are very specific for a specific persons. Okay, then next questions 32. A cosmid is a hybrid of what? Plasmid and phage. Phage is what? Phage is nothing but a virus which infects a bacteria. Okay, so hybrid of plasmid and phage DNA is called cosmid. Okay, this is used for what? DNA transfer, or in genetic transformations, or in what? DNA genetic engineering, we use this cosmid, okay, to create what? Transformants. Then 33. Optimum temperature of DNA polymerase T aquaticus is what? 72 degrees Celsius. So the most suitable temperature for Taq polymerase, it is also called Taq polymerase, is 72 degrees Celsius. Then base sequence of DNA can be determined by which method? Maxim Gilbert methods. We can utilize this method. Then 35. A particular protein in a mixture can be detected by which blotting technology? Western blotting technology. This is nothing but a blotting technology which find out the proteins by utilizing what? Gel electrophoresis, polymerase gel electrophoresis. Then all of the following statements about restriction endonuclease are true except what? 34. So except is they always produce sticky ends. So the restriction endonuclease, all restriction endonuclease does not produce sticky ends. They also produce what? Blonde end of the DNA sequence. Okay, there are two kinds of ends. One is blonde end, another one is sticky end. This is blonde end, and what is the sticky end then? This is sticky end. When they produce sticky ends means this is the sticky end. There is some portions is not same. Okay, so this is called sticky end, but it is not true. There some restriction endonuclease are also present. They produce a blonde end. Next questions. Suppression mutations occurs in where? Anticodon of tRNA. This is what? Suppressor mutations. Suppose a mutation occurs in a gene, but this mutation in the gene will be suppressed by what? Anticodon of tRNA. Okay, if there is another mutation that happens in tRNA for that particular gene, then there will be called suppression mutations. Okay, so right answer is D. Then in mammalian cell cycle, synthesis of DNA occurs during what? S phase. Okay, in S phase synthesis of DNA occurs. Then first protein synthesized by recombinant DNA technology was human insulin, this which is also useful for diabetes. Then lambda phage can be used to clone DNA fragments of the size what? Up to 20 kilobase, up to 20 kilobase. So next question 41 from microbiology and immunology. So the exact composition of a culture medium is known and it is called defined medium, because the exact compositions is known. Okay, next questions. Which of the following is ionizing radiations? So gamma rays are ionizing radiations and all other are non-ionizing radiations. So our right answer is gamma rays. Next questions. The growth curve obtained for a medium containing two carbon sources is called dioxic growth curve. So in this dioxic growth curve, what happens? Two different sources of carbon, or we can call glucose or galactose, both are present in one medium, and in that microbes are grown. This kind of situations or growth curve is called dioxic growth curve. Next, culture medium used for fungus is called what? Savarot medium, where we use fungus productions in a culture medium, then that kind of medium is called what? Savarot medium. Next questions 45. Which of the following organisms lack definite cell wall? This is mycoplasma. They don't have cell wall. Yeast is having cell wall. Mycobacteria, mucor, all are having cell wall, but mycoplasma doesn't have. Then bacterial cells are active and synthesizing new protoplasm. This stage of growth is called log phase, because here the bacterial cells are actively dividing. Okay, actively dividing, that's why this is log phase, means exponential phase, we also call exponential. Then 47, bacteria used organic compounds as their energy and electron source are said to be what? Organotrophs. So those bacteria which use what? Organic compounds as their source for food, source of carbon or source of food, then that is called what? Organotrophs. They are using what? Organic compounds. Then next question is 48. The antibody that is first formed after infection is what? IgM. IgM is the first antibody which is formed. Okay, this is pentameric IgM. Okay, antibodies in our body are produced by what? B lymphocytes. Okay, next questions. Active immunity is induced by what? Infection, not any kind of vaccinations or injections of antibodies. These are not introducing active immunity. This is only generated by help of infections. Okay, so in active immunity is induced by infection. So right answer is infections, because during infection the infecting microbes, the whole microorganisms will enter into the organisms and can cause an immune response immediately, and that immune response is called active immunity. Okay, next question. The specificity of an antibody is due to what? The specificity of an antibody is due to the variable portion of the heavy and light chain. If you take one example of an antibody, you will get this kind of structures. Okay, this kind of structures. So if you see this area are called variable regions, and the rest areas are constant regions, and the specificity is changed based upon the sequence followed in these variable regions. So the right answer is what? The variable portion of the heavy and light chain. Then fusion between a plasma cell and a tumor cell is called what? Hybridoma, and the technology is called hybridoma technology. Where we do this fusion between plasma cell and a tumor cell, we want to get antibody. So to produce monoclonal antibody, we fuse what? These plasma cells into a tumor cells. We will make the plasma cell into a tumor cell, by which what will happen? The plasma cells will grow very rapidly, and while growing, the plasma cell will also produce monoclonal antibody. So this fusion is done and the result is called hybridoma. Okay, next question. 53. Attenuated oral poliomyelitis vaccine is what? Sabin vaccine. These Sabin vaccines are attenuated. Attenuated means these vaccines will have viruses or any disease causing agents which are attenuated, means these are having non-infecting portions. Okay, that's why they are called attenuated vaccines or oral poliomyelitis vaccines, and these are Sabin vaccines. Then Mountex reaction is used for detection of what? We use the Mountex reaction to recognize TB, presence of TB. Okay. Then 55. During AIDS, acquired immunodeficiency syndrome, HIV infects which lymphocytes? CD4 lymphocytes. During AIDS, human immunodeficiency virus will infect the lymphocytes, CD4 lymphocytes, which are responsible for the immunity system of our body. That's why after AIDS, one person becomes susceptible to various diseases and very difficult to get recovered from that disease. So HIV infects CD4 lymphocytes. Then chloramphenicol is obtained from Streptomyces venezuelae. We produce what? Chloramphenicol by using which bacteria? That is Streptomyces venezuelae. Okay. Next, the etiological agent of botulism is neurotoxin. So during botulisms, the toxins released by the causative organisms is by nature neurotoxins. Okay. Next questions. Which one of the following mineral elements play an important role in biological nitrogen fixation? So magnesium is the important mineral element which plays a biological nitrogen fixations proper actions. Next questions. Penicillin inhibits what? Cell wall formations. So when we take penicillin, it will inhibit the cell wall formations of the causative organisms which causes disease in our systems. So what will happen? Cell wall will not be formed, and hence the causative organisms will not able to grow. Okay, so that's why this penicillin is an antibiotic. Then the reaction of soluble antigen with antibody is known by agglutinations. Okay, it will agglutinate or coagulate, and the process is called agglutinations. Okay, next questions from biotechnics and bioprocess technology. So the technique for purification of proteins that can be made specific for a given protein is called affinity chromatography. What is the technique? It's called affinity chromatography, in which we strain out the specific proteins. Okay, we strain out specific proteins by using affinity chromatography technology. Okay, next questions. Which of the following technique is used to separate proteins based upon differences in their masses? So we utilize SDS gel electrophoresis, okay, which separate different proteins based upon their mass. Okay, based upon their mass or size. Okay, so right answer is SDS gel electrophoresis. Next questions. In fermenter, the top portion left without broth is called headspace. Suppose this is the fermenter and we leave this much space. This is the liquid phase and we leave this phase without adding anything. This is called headspace. Okay. Next questions. The purification and recovery of the production after fermentation is called downstream processing. Okay, what is downstream processing? So when we in a fermenter, suppose this is a fermenter, and when we start producing some antibiotic, this is called upstream processing. But after the fermentation process is completed, when we try to separate our required antibodies or anything antibiotics, that process is called downstream processing, and in that process we utilize different technology like chromatography, affinity chromatography, centrifugations, HPLC, all these things, and these are called downstream processing. Next questions. 65. Microorganisms used for alcohol production is called Zymomonas mobilis. We utilize this specific microorganisms, Zymomonas mobilis. Okay, next question. Next question. Which method is not used to get immobilized enzymes? See, we utilize adsorptions, encapsulations, and what? Covalent bonding, all these things to get immobilized enzymes, but we don't, but flocculation is not used in what? Immobilized enzymes technology. Okay, this flocculation is rarely used, and this flocculation is used after the immobilized enzymes is developed, or when we try to get our product. Okay, next questions. Which of the following method is not followed to improve industrial strains? We utilize protoplasm fusion, recombinant DNA technology obviously, and genetic recombinations. All these things we utilize to improve our industrial strains, but de-replication is not used. Okay, next questions. The enzyme trypsin is specific for peptide bonds of basic amino acids. Okay, the trypsin enzymes is used to break peptide bonds of basic amino acids. So, next question. The specimen for an electron microscope is always stained with dyes, sliced into thin section, killed. It is viewed directly. Okay, we don't need to do the staining, slicing into the section, or we don't need to kill the specimen. We can view it directly by using one beam of electrons. Oh, okay, by projecting some beam of electrons into the specimens, we can view directly. Okay, next question. Proteins can be visualized directly in gels by staining them with dye. We can easily visualize the proteins by using some dye. Okay, next questions. Fluorescence microscope is a specifically modified compound microscope that utilize what? UV light. Okay, it can utilize UV light. Next questions. In gas chromatography, the compounds are separated on the basis of what? Partition coefficient. What they use? Partition coefficient. What did they use? Partition coefficients will be used in gas chromatography. So next is, in a native phase polyacrylamide gel electrophoresis, proteins are separated on the basis of what? Net negative charge. Okay, the proteins will have net negative charge while they are moving from one end to another end in a page, and the net negative charge will be active, and based upon that it will be attracted. Next questions. Which of the following range is associated with UV spectroscopy? In UV spectroscopy, we use 100 to 400 nanometer of wavelength of lights to identify the specimens. Okay. Next questions. Which among the following is widely used in Baker's yeast productions? So, the famous Saccharomyces cerevisiae is used in Baker's yeast productions. Okay, next questions. In the citric acid production, the pH to be maintained in the fermenter is 5 to 6 point. Actually, in citric acid productions, we need one acidic pH so that the growth of the microorganisms will be easy, and in that pH the growth will be proper and the citric acid production will be happen. Okay, next questions. The precursor for penicillin V is what? Phenoxytic acid. What is for precursor of penicillin V? Which of the following is not an impeller? Orifice sparger. Orifice sparger is not an impeller in a fermenter, because this orifice sparger is used to spray air bubbles so that proper mixing will happen inside the fermenter. So it is not a what? Question was to impeller. It is not an impeller. So in fermenter, the orifice sparger, which is responsible for the air bubbles throwing inside the fermenter, is called orifice sparger, and this is not an impeller. So the right answer is D. Okay. Next question. 79. Addition of nutrients, acid or alkali to fermenter is normally made through what? Silicon tubes. So we use silicon tubes to add nutrients or any acidic alkalis. Okay. Next questions. 80. The hydrogel obtained from natural polymers used for cell immobilization is polyacrylamide. We use what? Polyacrylamide to be used as hydrogel, and it is a polymer. It is what? It is a polymer. Next, 81 from animal, plant and environmental biotechnology. Which one of the following bacteria has found extensive use in genetic engineering work in plants? So in case of plants, we use what? Agrobacterium tumefaciens. This bacteria is used for development of transgenic plant. This agrobacterium tumefaciens, what they actually cause in plants, they cause what? Tumors, and during this tumor, the agrobacterium tumefaciens also changes the gene present in the plants. Genes is also changed, so it can develop hybrid or transgenic plant. So agrobacterium tumefaciens is utilized. Okay. So the right answer is D, is and very famous. So the medium used for selection of hybridoma cells is nothing but the selective medium, het medium, which is hypoxanthine aminopterin thymidine medium. Hypoxanthine aminopterin thymidine medium, and it is also called as het medium, and it is selective where only hybridoma cells will grow, and this hybridoma technology is only used for development of monoclonal antibody. Next question. Which of the enzyme responsible for cellulose degradations? So to degrade the cellulose, we utilize endoglucanase. Endoglucanase is used. Next questions. The cells have the capacity to generate different types of cells are called pluripotent cells. We know what is pluripotent cells. Those cells are also called meristematic cells. They have the capacity to pluripotent, so they can convert themselves to different types of cells. So that's why they are called pluripotent cells. Okay. Next questions. Which of the following bacteria is called well-eating bacteria? It is called Pseudomonas putida. This Pseudomonas putida is used for well-eating bacteria. In sea, there is a chances of pollution by the help of oil. So we utilize some bacteria called Pseudomonas putida to eat the oil or to degrade the oil and purify the seabed. That's why it is used. Then technique used to add oxygen directly to a site of contamination in the Beddoes zone is called what? It's called air sparging. What they are called? Air sparging. The technique is called air sparging. By this technique, we can add oxygen to that particular Beddoes zone. Okay. Next questions. Which hormone pair are required to differentiate callus? So what is callus? Callus is not a differentiated cells developed from a pluripotent cells. So we add particular auxins and cytokinins to differentiate their cells to our required types of cells. This is called auxins and cytokinins, and their ratio is also fixed for particular purpose. So we use this hormone pair. Next questions. Most widely used fusogens in protoplast fusion is nothing but PEG, or called PEG or PEG, and the full form of PEG is nothing but polyethylene glycol, and it is used as fusogen. Then gene knockout. What is the meaning of gene knockout? It is the process of inactivation of specific genes. So we utilize this gene knockout technology to inhibit cancer cells, or different techniques. Mutations, suppose a gene mutation is done, and we do what? Gene knockouts by doing what? Inactivation of that specific gene. Sometimes what happens, the activation of a gene leads to a disease, so we do gene knockout technology. By that, inactivations of the gene is happened. Then diabetes mellitus is due to the deficiency of insulin, one hormone responsible for insulins. By that, what happens, the glucose is not utilized, the level of glucose is not properly maintained in the blood, and the proper utilizations of the glucose is not happen. Glucose is supposed to be converted into glycogens, but it is not utilized. So the level of glucose is increased in the blood and leads to diabetes. Oh, okay. So insulin is used. Insulin is, if insulin is less in quantity or deficient in the body, then it leads to diabetes mellitus. Metabolic rate in mammals is controlled by what? Thyroid hormones. So if you see thyroid hormones, these thyroid hormones is known to control metabolisms, growth, and many other bodily functions. This thyroid gland, anterior pituitary gland, and hypothalamus are responsible for the various activity, but particularly if we check thyroid hormones, what it does, it increases the basal metabolic rate. What it does, it increases the basal metabolic rate, and it increases the gene expression of sodium potassium ATPase. So the more gene expression will happen, and what will happen, more ATPase will produced in different tissue, leading to what? Increased consumptions of oxygens. Increased consumptions of oxygen will happen because more ATPase is produced, and by that what will happen, respiration rate and body temperature is also increased. Depending upon the metabolic status, it can induce lipolysis or lipid synthesis. Thyroid hormones stimulate the metabolisms of carbohydrate and anabolisms of proteins. Okay, and thyroid hormones can also induce catabolism proteins in high doses. Okay, and thyroid hormones do not change the blood glucose level, but they can cause increased glucose reabsorptions, gluconeogenesis, glycogen synthesis, and glucose oxidation. So all these things are happened in thyroid hormones. Okay, so the next question is, the hormone known to participate in metabolism of calcium and phosphorus is nothing but calcitonin. This calcitonin is known to participate what? Metabolism of calcium and phosphorus, calcitonin. Okay, next question. Opines are amino acids derivatives found in bacterial cells. Opines are what? Amino acids derivatives found in bacterial cells. Next, the microorganisms leach metals out of rock ores is what? Thiobacillus species. So for leaching of the metals, we utilize Thiobacillus species. Next, the plasmids associated with the resistance to antibiotics are R strains. It is called R strains. In plasmids, we have one R strains, and it is associated to resistance of particular antibiotics, and is also used as a marker gene. And marker genes or the genes which can tell that these organisms, these microorganisms is having the desired plasmids. Okay, so it is called marker gene also. Then antisense RNA molecules have a sequence blank to normal RNA transcripts. So antisense of RNA molecules have a sequence of complementary to normal RNA transcriptions. This we can understand when there is a production of proteins by the help of antisense RNA. Okay, antisense RNA with the mRNA. Antisense RNAs are nothing but the tRNA molecules which are antisense sequence and which are complementary to what? Messenger RNA, and by that what? Amino acid sequences will be developed. Okay, next, which one is effective for the growth of shoot apices? So this hormones auxins is responsible for the growth of shoot apices in plant. So this is plant hormone and it is responsible for auxin, shoot apices development. Then asexual or vegetative propagation of whole plants using tissue culture technique is also called micropropagations. What is it called? Micropropagations. Okay, these questions already we have discussed. This is what? Callus. If it is unorganized proliferative cells, these are called callus, and we provide particular ratio of hormones to develop the callus into our desired way. Then the last question is, animal that has gained new genetic information from the acquisitions of foreign DNA is a obviously transgenic animal. So if an animal has or taken the foreign DNA into it, then it will be called transgenic animals. With this, we have completed almost 100 important questions from biotechnology. Thank you a lot.